Good evening and welcome to Crosstalks. Tonight we are live from Stockholm University, where we will engage in 3 different topics with a great group of distinguished guests. To those of you watching all over the world, you will tonight have the opportunity to ask questions of our guests attending here in the studio and over Skype. Just use your own Skype account and call us on Crosstalks TV, and we will do our very best to patch you through. What is the future of mankind in the universe? We ask a philosopher, an astronomer, and an experimental physicist. We'll talk about the role of cities in the societies of tomorrow, and we're starting the evening off with the revolution in the life sciences technology. With me to discuss the future of medicine are Professor Wouter van der Wijngaart from the KTH Royal School of Technology here in Stockholm, where he is leading bio, micro, and nanofluidics research at the Microsystems Technology Lab and is director of the Life Science Technologies platform. Welcome.
Thank you.
Professor Gunnar von Heijne leads the Center for Biomembrane Research at Stockholm University and has served as chairman of the Nobel Committee in Chemistry. Welcome. Jochen Schwenk is associate professor in translational proteomics and also manages the Biobank profiling facility at the Science for Life laboratory at KTH Royal Institute of Technology. Welcome. And joining us via Skype is Susan Lindquist, professor of biology at MIT. She's also a member of the Whitehead Institute and an investigator of the Howard Hughes Medical Institute. Welcome to Crosstalks.
Thank you very much. It's a pleasure to be here.
Thank you. I'd like to start by asking each of you, in your respective fields of research, what is to you the most exciting frontier? Let's start with Gunnar.
That's a good question. So my own research deals with molecules inside cells. I think, though, that the most exciting life science area right now, and certainly for the next 10 years or 20 years, is neuroscience. What happens in neural cells, what happens in our brains, how do we describe that? And it's a very interesting two-prong approach, because we have all the molecular sciences dealing with what happens inside neurons, what are the molecules, how do neurons connect, on the one hand, and then on the other hand we have psychology, who is dealing with, the people there deal with the brain, characterizing how the brain takes in information, processes information, interprets information, and places us in an environment somehow.
So we're in a situation where the sort of natural science world will is very soon or already applicable to everyday life.
I don't think we're there very soon, and that's what makes it so exciting, because there is this big rift between the top-down approach of psychology and the bottom-up approach of the molecular sciences. And I think we're still a far cry away from merging the two, right? And I don't know, Susan, you're much closer to neurobiology than I am. I don't know if you would agree on this, or if you have a completely different view on things.
I think I agree with you 100%. We're at an extraordinarily exciting time. It's an absolute frontier in every direction we look. The excitement of understanding how a brain works, how thought happens, how these extraordinary brains we have process all the information and create new knowledge. It's just really quite remarkable how we learn. But the area of neurological research that I have been delving into lately has been what happens when it goes wrong. So that's actually also becoming an exciting frontier now. Neurodegenerative diseases have been such a scourge of mankind, and as we have lived longer and longer lives with all the advances and remarkable advances of science in the last century, we've doubled the human life expectancy. We're now living longer, and the bad side of that is that neurodegenerative diseases are waiting for us out there. And when I first started taking what was a very basic science approach to understanding protein folding and starting to see, well, can we understand what's going wrong when these proteins are clogging up the brains, and can we make some headway in that? At first, I really found it so depressing, because it seemed that it was just going to be forever before we'd ever solve the problem. But with the new tools of biotechnology and the new insights on the genome and new levels of understanding imaging of the brain and how neurons are connected to each other. Those basic frontiers are actually helping also to empower, I think, a new way of approaching this very, very large problem that will be an economic disaster for us if we don't solve it.
How about Jochen?
Yeah, I think my view is really using, for instance, the name of the facility that I'm working for. So Biobank profiling, so meaning Biobank is a collection of many samples from a population. So to really involve a population in research, to make it aware of activities within research, but also to connect with other scientists, epidemiologists that understand, diseases on a more global scale. I think neurodegenerative diseases, of course, are very important since we, as Susan said, grow older. Cancer is, of course, always an issue, cardiovascular diseases. And then using the technologies that we are working on in the lab, using the reagents that are made within the project that we connected to generate new information and hopefully at some point to find new clues about how to diagnose a disease early, to be able to treat it, to understand it better. And of course, since we use the blood as the source of information, that is connected to every organ, so we have some sort of more systemic view on a human being. So that's, of course, a great challenge to understand that and connect it.
This is
I think one of the most extraordinary things that I've witnessed in my time in science has been how this global community has come together and how engineers and clinicians and biologists and just people who used to work kind of very separately from each other are now interacting and creating, really new synergies and excitement in ways that as a human being doing science, I find that very exciting too.
Wouter is nodding frenetically.
Yes, very much. And it's one of the big challenges also too. I think that engineering and medicine perhaps have grown separate for a long time, where engineering gets more and more specialized in technical high schools. And we really have to find the way back and interact with the medical people to understand how can we use technologies in medicine and how can we, that would be one of the major challenges we're focused on.
I think what you've all established here is that it's almost like peeking over this hill and seeing this vast landscape. And I think we need to sort of narrow down a little bit more what's happening right now. So if we just go a little bit back, Gunnar, could you just briefly describe some of the major science breakthroughs that have led us to this point? And then let's try and see what everybody's doing to see where we are now.
Of course, we're in the midst of an explosion in biological knowledge. That's for sure. And a lot of it is because of this merger between technology, biology, computer science, and physics. On the other hand, I think we have to be a little bit humble and look back, because the really big breakthroughs in biology, I think, so far, date back, more than 100 years. Mm-hmm. And of course, I'm thinking about the theory of evolution, Charles Darwin, 1859 or something, Origin of Species. I mean, that is the one humongous breakthrough in biology, I think, that permeates every aspect of biology as we understand it today. Mm. So that's absolutely the keystone to the whole edifice of biology. And then along the way, there is another big breakthrough, which is the nature of the genetic material. How can genetic heredity be passed on from generations? And that is, of course, the discovery that DNA is the material basis, the molecule that encodes the genetic information, and epitomized by the discovery of Crick and Watson, their model for the double helix of the DNA. So in some sense, although we have all this fantastic, sophisticated technology, and we're producing all this amazing knowledge now, in a way we're just kind of trying to work out the implications of these really big breakthroughs. It's 60 years since the DNA molecule structure of the DNA was proposed, right? And I think we're still just dealing with the implications of that, although we're doing it in much more sophisticated ways than what we could before.
Then let's jump to something that's being done right now, because Wouter, you've brought us something. Could you just show us what you got there? Something that's so recent that as of like yesterday, we couldn't even show it because it didn't exist.
No.
What have we got there?
I got it yesterday in my office. It's built by my PhD students. So. This is a device we've manufactured in our lab. We work with microsystems, and this device is actually it's a second generation prototype, and it's going to become a measurement tool, point of care measurement tool for influenza. So the idea is that patients will breathe in here, and we'll detect whether they have or they don't have influenza.
What does it just from the breath?
Yes.
I can see how that would be very useful for me when I travel, if all the other passengers on the airplane would be checked before I go on board.
Yes.
So how does it work?
I can't tell too much about that, I'm sorry, but how it works. But indeed, it has a number of application areas. And one is, of course, a patient at the doctor's side. Another would be screening of people in big rooms or entering airplanes. Another would be in the case of pandemics. But this kind of technology is really, I would say, driven by how much are you willing to pay for a test like this. And that really depends from situation to situation. If you have a normal flu epidemic, then if you come with a cough to the doctor knows you have influenza, he doesn't need to measure it. He doesn't want to pay for this technology. But if you would have like the pandemic of influenza where 1/3 of the people died, and everybody wants to have it, of course. And so it's really what is the cost economics behind such a tool and how to use it, and that's driving the technology.
But the technology exists now. More or less.
No, this is a prototype. It's not working yet. I wish it was, but.
Okay, when can I go for my breathalyzer flu test?
Well, that depends indeed whether you have a pandemic. Or if there's a pandemic would be starting tomorrow, then I think we'll get a lot of money in this project. I think we can speed up the research quite a lot.
See how this is working now.
Yes.
Yeah. We were talking about DNA, and of course everybody's, even laymen like me have been following the project that has to do with DNA mapping. But now DNA seems to be a little bit yesterday's news. And Susan, I would ask you, just for those in the audience who perhaps think of protein mainly as steak, very briefly, what is a protein?
Right, but proteins, we eat proteins because we need to make our own. We need to break them down and make our own. Proteins are basically us. The DNA is the code for life. And for those of you who out there who can remember cassette tapes and tape players, it's very much like the tape. It very, very complicated instructions for life. But it actually itself looks pretty simple and not all that interesting. But when you put it into that tape player, this extraordinary complexity and richness of music comes out of it. And that's basically what's playing the tape. And what is the music itself is really proteins. And so our DNA codes for thousands and thousands of different proteins, and proteins power our muscles, they digest our food, they harvest the light in our eyes and send signals to our brains so we can interpret it. They basically do everything that we think about as part of our lives. And getting that code of DNA decoded into functioning thousands and thousands of different proteins, that's really what's been driving my research for the past 25 years or so.
So we know it works most of the time because here we are, but we don't know exactly how.
Yeah, it's really quite extraordinary. You can think of the nature of the problem sort of as if you were to try to assemble a musical orchestra, and you started out with a whole bunch of sheets of metal, and you had to fold them very, very precisely and mold them and bulge them out in certain places in order to make a musical instrument, and you had to make lots of those musical instruments. And then the way life works is that they're all crowded together in an incredibly dense space, and they're moving around all the time, and yet they have to this orchestra has to work and produce this extraordinary thing which we call life. And so quite often, unfortunately, that process winds up getting a little chaotic and going astray, and that can happen just with aging. Proteins start to misbehave and clump up and not do what they're supposed to do, or it can happen with genetic mutations. As we were talking just a moment ago, having this bank of tissues from all these human beings now, we're sequencing these genomes and finding problems in the DNA code that can lead to this orchestra not working.
So would that be?
Pardon?
Sorry, would that be, you mentioned protein folding before, so if it folds wrong, is that what happens
Yeah, you can
when
imagine
protein
if you have
breaks?
even one musical instrument in an orchestra, that the person who was making that trombone didn't get it quite right, and it could really ruin the whole orchestra. That basically is a way in which mutations in genetic code can lead to terrible diseases. But even just wear and tear and aging can lead to problems in those instruments. And really, probably about half of all major diseases that beset mankind, and we mentioned neurodegenerative diseases because Gunnar opened with this beautiful frontier of neurobiology, but most cancers are caused by mutations that cause proteins to not be behaving properly, and they signal let's to the tumor, let's grow at times when they shouldn't. Many infectious diseases are actually very, very tied closely to the realm of protein folding world as well.
That's really interesting. Jochen, you're also part of a large international team working on the creation of a human protein atlas. So the aim is to systematically map the human proteome. Can you tell us why this started? I mean, we have a clue now, from Susan, of course. And what development, what discoveries has this project made since the start?
So, I mean, of course, it started as a follow-up to the work on the human genome, which was first drafted, I think, in 2001. So, and following to that, of course, the information that was generated led to, of course, new possibilities, and that is to map out all the proteins that are actually encoded in the genome. And I think it was about 2003 that Matthieu Salin sat together with colleagues and thought about, okay, how can we systematically approach the discovery or the analysis of all proteins in the human body? And that is basically by producing antibodies against the protein encoding genes. So by 2005, the first version of the Human Protein Atlas, which is a public portal that everybody outside this room and in the whole world of science can look into where proteins are expressed in the body, in which tissue, and eventually to investigate how protein expression changes, for instance, in disease such as cancer. So of course, one of the major questions is every protein expressed in every cell? So at the moment, our understanding is that the vast majority of proteins are actually expressed in every cell. So there are very few tissue-specific proteins that are found. So of course, that then leads to following questions. So what does it mean mechanistically? So every cell has a certain function. So, and of course, what does it imply for diseases? And I really like Susan's analogy with the orchestra, but maybe I want to add one because that's what I usually use when I talk to students, and especially friends outside science, and is using the analogy with medieval cities. You know, you have the wall, you have kind of the inner city, you have church, and in the basement of the church, there's a plan for the whole city, how it's supposed to be built, where should be the bakery, where should be the mill, where should be, I don't know, that gate, that tower. And I think just to see the DNA as this plan, and somebody has to go to the DNA to read it and to say, okay, we need bricks to build this hospital, or we need to build a new power plant, or mitochondria, as science calls it, is of course fascinating because it means communication, interaction, and who tells who what to do what. And I think this is really, of course, not a static information, and I guess many agree that the genome is not static either, but of course the proteome, which is the sum of all proteins, is of course much more active and changes, and is of course also responding to the environment a lot.
Right. You can imagine if that medieval town every building was actually on wheels, and they were all moving around. It's not just the people moving around, but all the buildings were moving around, and the walls were being moved, and so life is just this incredibly active environment where making things happen. Part of what drives life is the fact that it's so active.
I'm starting to understand that even the process of thinking about these things has to do about zooming into the sort of right distance mentally. Gunnar von Heijne, your work combines computational biology with cell membrane proteins, and I'm starting to understand that the mass of things happening here is so complicated that you might need some big computation tools. So what kind of things is your laboratory bringing to the table? What kind of knowledge?
So I think I can connect to this idea or model of the cell as a medieval city with a wall around it, and we're particularly interested in the proteins, you know, Susan's proteins, but the ones that sit in this wall. You could say the gate proteins, the guys that let things in and out. And we're very much basic science. We're just trying to understand more and more about these wall proteins, these membrane proteins, and how they get, how do you put a wall in, how do you put a gate into the wall? You have to open it up somehow when you put the gate there. That's one area that we're trying to understand. And in doing this, we can also start using genomic information or the protein information that something like the Human Protein Atlas brings to the table, and trying to predict aspects of these gate proteins, these membrane proteins. What do they look like? How do they fold into a defined shape, and sometimes misfold, as in Susan's case?
Okay.
How do they work?
This is fascinating, and I do understand that research is inevitably very often focused on the next publication or the next funding deadline or something like that. But if we try to go back to that sort of view of the whole valley, what would you say is the ultimate goal here of life science research in general, and especially, of course, I guess the tech side? I mean, this is a big question, admittedly, but what are we trying to achieve here?
Well, I think that by understanding how life works, and this, first of all, it's the most extraordinary intellectual question I can ever imagine being privileged enough to engage in. It's just fantastic. How does this life work? But I think, from my viewpoint, really what I see amongst my colleagues is this real determination that through this extraordinary process of discovery, we're really going to be able to solve some of the really terrible problems that mankind is facing. And I think that's really, that really is on the horizon. I mean, developing newer, better vaccines. Infectious organisms are we've conquered them briefly, but that's not necessarily going to last for long. The capacity of biological systems to evolve doesn't mean that we're really safe from infectious disease, but it's not. Every sort of aspect of human biology and development, giving every person on this world who is born at some time, we will have, I think, within our grasp, the capacity to give them longer, healthier, and more fulfilling lives.
Okay, I read a lot of science fiction. We're not talking about eternal life, though.
No.
But longer, yeah?
Yes.
But, Wouter.
Well, I don't know if it's indeed longer life. This is actually a political question that you have to ask. What is good life, and when we look in medicine, what is the aim of medicine? If I talk, I hear many people describe. I don't know if this is an official view of the state. I don't know if there's such a thing, but I hear many people say, yes, we want to have a long life, and then once we get sick, we want to go fast and painless and have a quick death. So maybe, is that an ideal life? I don't know.
But is it, I mean, do you guys ever think about these things? Yes. Do you think about the longer perspective? What are you working for? Yeah, Jochen's nodding. Yeah,
absolutely. I mean, this is what I think all of us drives us in the back of our heads, even though it's not so obvious on a daily basis. I mean, if we look at protein sequences, structures, or at building microsystems, of course, that's not really like, oh yeah, what we want to aim for. But it's taking the small steps. But sometimes, I don't know, if you take a walk on the beach or in the forest, you think about things. Okay, what am I actually doing here? And sometimes, of course, that's okay. I'm working on a good thing because I'm trying to help to understand, to provide a little piece of the big puzzle that we all are looking at. And I think I want to pick up one thing that Wouter said, and it's discussed, is of course, what is healthy? I mean, what is life quality? And who's normal, right? And I think this is a very important aspect, especially for my research, because we usually deal with people that are not healthy, that have a specific disease, but then we compare them to some other people that, in this regard, are considered as normal or healthy, but are they? Is this really something that's important? So what makes the fine difference between healthy and diseased, and when can we define somebody is not healthy? And I think at least our work, and I mean bringing together different people from different aspects and perspectives of science, is really the challenge to make something that improves life quality, because that's, I think, the aim of everyone, to have a good life and to have a life that you can live.
This raises.
I think one of the things that to actually turn back to something Gunnar said earlier was that evolution happened and life evolved from simpler forms, and our understanding of that process has really just grown extraordinarily. And one of the things I'm trying to do is to recognizing that this problem of protein folding, which causes so many diseases, is really very ancient. It's part of the initial fabric of life, and really harnessing this new knowledge of evolutionary processes that we've gotten from genome sequencing to understand how the protein folding problem can plays out to so many different types of diseases, and connecting with all of these different technologies, I find that it's really present in my mind all the time. You know, what eventually can we use this knowledge to help people live better lives? And I think that often, you know, there's a lot of scientists being portrayed in a lot of different movies in which we're mad and want to control the world, and it's truly not the way I see things in science these days. I really see my colleagues and my students and all over the globe when I travel and go to meetings, people really care about making a difference in this world.
Gunnar is laughing and nodding.
Yes, no, I mean, I definitely agree, but there is one sort of interesting aspect, I think, of science, which is that even if you're not driven by, you know, by bettering the lot of mankind or something, even if you're only driven by the intellectual curiosity, in the long run, your work is going to benefit mankind. And I mean, that's a great invention of science, actually, that you can funnel even just curiosity-driven research in this way.
But now we're saying mankind. Do we really just mean sort of the global north and whoever can afford it? Or are we talking about once this knowledge is out there, we're going to be able to use it to counteract social injustice and these kinds of things? You're all nodding now. I was thinking you were going to say, no, unfortunately, this is only for rich people. So how is this going to help everybody?
I mean, the world is growing in its a number of people. So I mean, that not only means that there are more to take care of, but also that we are moving closer together, just simply because we expand in size. And now with modern technologies, with transportation, things are not like so stable in terms of that they stay at one specific region or area. So we all, I think, are connected somehow. I mean, I think the influenza that kind of broke out a couple of years back clearly showed you cannot just say, oh, those people, you know, over there, they have this and this problem. That will not happen to me. So I think we should be, you know, caring about everyone in a sense. You know, of course, it's not possible, but at least
technology has a big role to play here. I mean, we talk about life science and you talk about biology, but on the other side of life science is medical technology. It's a really big part of life sciences. And actually what we're trying to do a lot within medical technology, for example, with the microsystem we're building, is really bring the medical technology to a very low cost, so that it can really that the inventions you're doing in SciLife Lab can be brought to medical practice at a low enough cost that you can have it actually also in a third world setting.
Gunnar.
Right.
Just a moment, Susan.
Interesting. More people on this earth have died of malaria than all other diseases combined, and it's one of the great killers in our modern world, and it affects primarily the disadvantaged nations, not our northern developed world. But one of the exciting things is that in this town where I am working, I'm connecting up with all kinds of people who are studying malaria. And what their goal really is to do, and it actually, I intersect with them just a little bit because of some of the issues of protein folding, but one of their goals is to be able to cure malaria with something that's going to cost pennies, because unless you can get a dose of an anti-malarial and manufacture it for really the cost of a dime or so, it's not going to help the third world. But people are really focusing on trying to do that.
In the last few weeks, at least here in Sweden, and there's been a lot of media attention around biohacking. So that would be interested amateurs in home-built laboratories, getting into DNA analysis, virus design, and so on. And I guess this is based on the idea that biological programming is not very unlike computer programming, and that these skills should be available to all. How do you guys feel about this amateur competition? Gunnar.
You know, what this brings to my mind is, you know, when I was a teenager, those of us who were geeks, who were nerds, science nerds, we had a dark room in the basement, and we were doing, you know, developing films, and we were mixing chemicals and stuff. It's a bit similar, I think. You know, it's today's nerds, and it's great. It's great that we have these nerds that do these things. I'm not sure if if biohacking, in principle, they can do more things than we could do in a dark room, both good and bad. But I think it's today's teenage nerds, basically, and they will be good scientists in 10 years' time.
They're going to be good scientists. Susan.
I can remember that when I was a teenager, I desperately wanted a chemistry set. I wanted it so much, and as a girl, I was not given a chemistry set. I was given instead a little baking set. But I really do feel that having people be able to engage with what life science is, having young people around the world being able to think about biological experiments and be excited about them, I think it's to me, I don't think of it in any way as competition. I think of it as a very exciting way to engage people in this glorious thing of how life works.
I have to say, of course, it seems from the coverage that the national security organizations all over the world are quite worried about this. But
I think it is. I think that's not without foundation. That there should be some concern for that. I'm more concerned about a determined bioterrorist wanting to use this knowledge and wanting to tinker than I am really with teenagers in their basements.
So really the problem is, the risk is really from people who have actual knowledge rather than people who are making it up as they go along.
I think it is, but there are unexpected things that can happen. But that's where I see the real danger coming from.
The unexpected things that can happen, and again, I've been watching a lot of movies, but I do have to ask you this question. Is there a real risk that you guys or somebody of your colleagues somewhere is going to accidentally, or even worse, on purpose, develop something quite lethal and quite dangerous, and then that might get stolen or might spread? Or do you feel that you're part of a brotherhood of science or sisterhood of science where you have very strong ethical guidelines and you would absolutely never research anything dangerous?
Well, I think it's important to recognize also that biology is out there doing this all the time. I mean, HIV appeared out of nowhere, right? It was a new epidemic and a terrible scourge. And that happened because organisms were contacting other organisms and exchanging information and exchanging DNA. And this is how life has evolved on this earth. And so I really think an awful lot of stuff has been going on. And the more we understand how to control that and develop therapeutics for this, I think in general, it's the more we know, it's going to be for the better. I dearly hope and believe that.
I'm still not sure that you should be helping nature along down certain paths. Wouter, what do you say?
Yeah, isn't it so that 10% of the human DNA has a viral origin, or?
Yes, exactly.
Yeah. So
can you translate that to a layman? What does it mean? 10%?
That means that, for example, I don't know, it's not my specialty area, but if you are exposed long enough to HIV as a human race, I mean, it's going to be become part of our genome, and you don't see it as a disease. It's just part of your natural genome, and I think that's how evolution works, basically.
Yes, that's one of the ways in which evolution works. This exchange of genetic information and viruses hopping around from one organism to another, and there's really an extraordinary amount of biology that's happening out there all of the time.
Actually, one major threat I would say is not that we're seeing now is not people trying to make viruses, but it's antibiotic resistance, and us in industry, for example, feeding antibiotics to our cows, that then end up to antibiotic resistance that is spreading. And that is a real big problem nowadays, antibiotic resistance of gram-negative bacteria.
Gunnar?
Yes, no, I agree. And
that is bioterrorism. No, I don't know.
In some sense. Nature is much better than us at inventing these bad, for us, bad things. Yeah.
But there's still something about this, like, that there are certain usages and things that are sort of becoming systemic and normalized. I mean, genetic selection in children. We're again, it's about what is normal, but we are making these kinds of choices increasingly where it is available. It's of course frowned upon terribly and with good reason when the method is selective abortion. That's absolutely horrible. But a lot of the same people who are judging this or condemning this are looking with great interest at the possibilities of selecting away certain traits in their own children at an earlier stage.
I think that the truth of the matter is that all progress comes with there's associated good and bad with it, and what's frightening to people now, I think, is that the pace seems to be accelerating so much more. I mean, the rate at which our lives are really different from the lives our parents led is extraordinary, and my children are now using electronic technology that I can't figure out how to work. So, I think that's happening all over, and our lives are being transformed. And one has to really have hope, but also be as vigilant as we can be, that we use it for the best possible purposes we can.
Okay, in that case, can I ask you a really personal question? you don't have to answer, but like, have you ever been at a moment in your own research where you've looked at something or realized something and gone, wow, this could really be used in a bad way as well? I'm not going to go there, but it might. Have you been in that situation? Gunnar is nodding. Yeah, Susan as well.
Absolutely.
Yeah.
I mean, if you would analyze a sample of a patient and you find something that, at your kind of the moment you look at it, excited by the discovery, you know, extrapolate, this could mean that this person has this and this disease. Of course, this is frightening in a way because you have yet no proof. Mm. So I think I think every knowledge is linked to responsibility. So if we have the knowledge of today, like you said, you know, if we look at our children before they're born, to kind of say, okay, we want to have them because they are kind of bulletproof in terms of they have every piece that they need to grow up and be an adult. I think that comes with a responsibility to see, okay, what does it really mean for us and for our society and for the world? So I think it's not only just generating knowledge, but it's also, you know, seeing the value and the power it has.
I think we have a question now from a Skyper in Pakistan, I think. Do we have you online? Hello?
Hello, yes, I can hear you. Can you hear me?
Yes, we can hear you. What is your name, please?
It's Rana Munawar Farooq from Pakistan, and basically I'm from the field of education. I'm principal of the school, as well as I'm a student of political sciences.
Welcome. So what is your question?
My question is this, that we are having many Down syndromes in our societies. So can we cure these Downs before pregnancy or after birth?
Okay, thank you very much for this question. So what are we saying? Down syndrome. I have to say that there are, of course, many people who feel that this shouldn't be, that this is a condition that doesn't need curing, but there are, again, a lot of suffering connected to it that many parents certainly would want to prevent. Can we cure or prevent Down syndrome? Gunnar is nodding. No?
As far as I know, we cannot cure it, but I may be wrong. It can be diagnosed, of course. At early during pregnancy, but
I mean, it means that we interfere in a process that is so powerful. I mean, the growth of an embryo into a small baby is, I mean, such a powerful event. So many things happen, exploding in terms of the size in a very short amount of time. So the question is, if we would change a small little detail, what would be the consequence? And I think it's very difficult, at least from my understanding, to predict what the outcome would be.
So exactly, I should say what we're talking about here, of course, is chromosome right. Design, right? And that's not going to be happening anytime soon, nor perhaps should it. Susan?
The problem with Down syndrome is that there's the embryo gets started with a whole extra large chunk of genetic information. So it's really an incredibly complex disease. There are 1,000 genes on that chromosome, and so it's not just one protein going wrong, it's an imbalance between hundreds and hundreds of proteins. So that is just something we can't begin to tackle at the moment. And whether we ever could, I don't know. But this is where things cross over into diagnostics and difficult questions of whether abortions should be allowed, difficult questions of whether earlier and earlier diagnosis or just informing people of the risk. This is hit on a really difficult biological problem and therefore also a very difficult ethical one.
Yes. Thank you. I'm also opening for questions from the audience, if anyone should have one, or do we have another one on Skype? How are we on that? Here, please, move to the microphone, please, and state your name. Yes.
Good evening, everyone.
Good evening.
My question is, I'm a master's student. When I look at the peer review journals, the best ones in the business, I get to see a lot of exciting stuff that's being published in these papers. But my question is, what are the challenges that scientists are facing to to take these interesting ideas and their inventions on a commercial scale? And what are the difficulties in taking them into a commercial use?
Thank you. Thank you for your question. Who wants to begin? Wouter?
Well,
you're sitting there with the prototype, so we start with you.
Well, I think, for example, you investigate a new biomolecule, a biomarker, and you see it has to do with one or another disease. But I think biological processes, as you have mentioned before, are often so complex that changing one marker that you find, and you have to understand what is the entire role of this marker and how does it collaborate with all the other biological processes. And this is where it very often very quickly fails, I think, because it shows up. It's never only one marker. The world is so much more complex that you very often you fail immediately while trying to use that marker for something. I think that's a major failure point.
The transition of science in academic laboratories and make it really matter out in the real world. I think it's a brilliant question because it's really one of the great dilemmas of our time. I think the funding mechanisms that we have had traditionally in place to do this, with venture capitalists investing in what looks like a good idea, and that idea usually plays out in a couple of years, and the company either works or it goes bust. Well, a lot of the ideas that need to be translated now are going to take a much longer time frame to get them translated. And so the dilemma of how do we take this new knowledge and make it really matter in the real world in commercial products is an extraordinarily important one.
It seems to me that the expansion is such that it's clear, and also, of course, the need for resources for testing and so on is so vast that it's not going to happen in academic context. Basic research, I hope, will still be funded, but a lot of this will have to be funded commercially. Jochen, you were saying?
Yeah, I think the sad aspect is there needs to be a market for something like this. I mean, whatever the discovery is, if there's no market, nobody will buy it. So then, of course, every brilliant idea is lost halfway. Another problem is, of course, that discoveries are often made on a small scale in an isolated environment. Let's say a certain population is being screened for a certain disease, and of course, the excitement really arises because it's a new finding that's connected, for instance, a genome to a disease status. But of course, if we would put it on a more global scale, we need to involve more players, more ethnicities, more people. And of course, that is usually, at least in my field, kind of the real challenge that people face. It's discovery is easy and exciting, but verification is so difficult. And of course, that is the challenge because that needs new technologies, needs a better understanding of the molecules, how they fold, how they interact. And I think then to integrate all the different disciplines into achieving a better understanding and eventually something that mankind can benefit from is key.
I'm going to ask you a question now that may be unanswerable, but at what point do you think a researcher should leave the academic sphere? Or should you stand with one foot in both always?
Stand with one foot in both. Basically, if we do research, I mean, either you have a great scientific paper, but if you want to bring that further, either you have to go to technology, you have to either get a patent, or you have to out-license the technology, or give it for free to a company, or you have to start a company yourself if you want something to do with that. And I think we have an obligation to do that, to bring it a step further.
I think more and more academics are doing that. I started a company myself, and it was a successful one, but it was really not easy. And I think academics need to gain an appreciation of how translating something into a real commercial world and something that actually works, and the complexities of trying to find a compound that will fix a certain disease in a test tube in a dish in a laboratory versus actually getting something that works in a human being and it lasts in the body and isn't excreted and doesn't have extra toxicities. It's really an incredibly complex problem, and I think trying to understand, I think it's really been a transformation in my professional life in science. Has been when I started out in science, I never even dreamt of trying to do something like that. Now I think more and more academics, at least the ones I see around me, are really trying to make that effort, and I think trying to find ways that will ease that path and educate them in how best to do it is going to be a very important aspect of
science education as well. Gunnar?
Yes.
I'd also like to put in a plug for basic science. I don't think every scientist has to have one foot in each camp. I think some scientists should. Some scientists should be more on, the applied side.
It depends on your personality.
It depends on personality, depends on interest and everything, where you can make the greatest difference, But we have probably been separating the two camps too much, and they need to be brought together, but not each person.
It does seem that the sort of questionability, it used to be if you're in please go ahead. It used to be that if you're involved in a commercial venture, you're becoming somehow suspicious as a scientist. That's disappeared entirely, I feel. Yeah. Yes, please.
Thank you. My name is Mario Romero. I'm from KTH, and thank you for organizing this. I want to veer the conversation for maybe a few minutes towards autism. I'm reading the CDC report from March this year, and the prevalence has increased to 2%. So that's 1 in 50 children, 6 to 17 years old. If you consider that autism is a lifelong condition, and you also consider that depending on the severity, there may be many adults per child dedicated to that child's life, and then what happens after that child reaches adulthood, it's sort of a big, what Wouter was saying, it starts to look like an epidemic. So the question is, well, really, I just want to sort of have a conversation, but the question is, or one of the questions is, what is the advancement in biomarkers for autism and early detection and intervention for autism? And why is the prevalence going so high? Is it a matter of a change in diagnosis? Is it a political matter to get access to resources from insurance? Or is there a combination of genetic and contextual factors that are triggering the disease?
Thank you, Mario, and thank you for the sort of broad approach on this. And of course, now, if none of you have a specific specialty in autism, you feel free also to approach this question on a sort of higher level. But immediately, if we start with it, does anyone have?
I have something I could say about it. I think there's no question now. The thought that maybe we're just diagnosing it better was out there for quite a while, but I think there's no question now that it is on the rise, it is increasing. And we don't understand why. It may be because of, and is very likely, an intersection between environmental factors, potentially some viruses we don't know about. It's a great question that is occupying a very great many people right now. And I think an example of what I find an inspiring sort of approach on this is there's a center here at MIT which was funded recently by a philanthropist who made a great deal of money. He's very concerned about this problem. So he set up an institute here to study autism. And what I find really inspiring about the way that has been done is that they're not only trying to find biomarkers and figure out how you diagnose it, they're really approaching it from the standpoint of the basic science problem. You know, what goes wrong when nerves are not connected to each other quite right, when they're not talking with each other quite properly, and they're even using very, very simple organisms to try to probe what's going on. And I really love the fact that it's bringing together people from all sorts of different disciplines, the geneticists and chemists and brain scientists. And I think that's an example of it. It also connects to the question that was asked about how do you translate technology. And I think sometimes having someone with vision who says, I'm willing to fund this high-risk, potentially high-payoff endeavor amongst a wide group of people who are doing different things. That doesn't happen all the time, unfortunately, but it can happen, and I think it's inspiring when it does.
Very briefly, do any of you others have input on this? Dare to speculate?
It's of course a good example of something becoming very, very obvious and very kind of, let's say, challenging to face, because when do you kind of start looking for a marker? How do you start looking for a marker? Which are the tools you want to use? I mean, there's so many entry points into this question and into kind of finding the right solution for it that it's of course challenging at one point, but I think the most important thing is that the first seed is planted, that there is a problem we need to face and we need to put our heads together and we need to find the best possible solution for helping those children, or for helping parents that are planning to have children, or look into certain environments. I think that there's so many aspects that of course need to come together and to find the right strategy for it. So I think the important thing already happened, that people are concerned and observant. They say, okay, this is happening, we need to do something about it, and then of course there are gazillions of ways of approaching it. And I'm not an expert in this field either, but I can only hope that the smart heads in the world find a motivation to build smarter groups for tackling that disease.
And it does seem that a lot of the things we touched upon, that is going to be the answer, getting people from different fields working together. We're running out of time, so I have one final question. Could each of you mention one problem that humanity is very close to solving. Let's say, I don't know what very close means in your field, but perhaps, let's say, maybe 10 years, something along those lines. Very, very briefly.
From my point of view as a basic scientist, I think it's kind of an ill-posed question. Is what we consider a problem today, we will not consider a problem tomorrow. And you know, the whole idea of the problem is evolving all the time. Mm-hmm. So I don't think we could, you know, even put a, you know, stamp on a timeline and say, okay, problem solved. Because by that time, the problem is different.
After this answer, I'm not sure it's possible to answer any of you. Any disease? Yeah, Wouter, briefly.
I talk from a medical technology point of view, and I can see some of the trends coming within the next 10 years. And talking about my own field of microsystem, we're coming to a system where people will be able to diagnose themselves more in the home setting, and so that's going to change perhaps medicine and make it more cost efficient and maybe better. So these are definitely trends that we're seeing, and that's going to change.
Right. I think there's been a lot of hype about personalized medicine, but I do think it's real, and it's going to be empowered by not just being able to sequence our DNA. I mean, it cost $1 billion to sequence the first human genome, and now it costs a few thousand.
In that case,
but that's only one of the things we have to do in terms of identifying what's the basic cause of certain diseases. Then you have to find the chemical compounds that are going to combat it. And I think that is a new horizon. It will lead to some very exciting new things in the future in the next 10 years.
Thank you so much. Wouter van der Werenhart, Gunnar von Heijne, Jochen Schwenk, and Susan Lindquist. We'll be back on the hour with the cities of the future.